Hello, my name's Olivia Gearner, and I'm going to be talking to you all about some of the research I've been doing on the evolution of substrate tapping behavior in the African Tok-Tok beetle. Tok-Tokis are darkling beetles in the tribe Sepidiini, which is a familiine tribe present in Africa and around the Mediterranean. Like most familiini, they are found in deserts and other arid and semi-arid habitats. This tribe is very large and morphologically diverse, containing around 1,000 described species and subspecies. Many species in this tribe are known as Tok-Tokis due to their substrate tapping behavior, in which the males tap their abdomens on the ground and the females tap in response as a form of sexual communication, which you can see here in this video. So there, the male was tapping and waiting for a response from a female. It's been hypothesized that this behavior conserves energy and water by reducing the amount of searching males need to do for finding mates. And so far, only members of the subtribe Molarina and the former subtribe Phaneratomina are known to do this tapping behavior. So the research questions I sought to answer are which species tap, and are there any outside of Molarina that tap? Do taps differ between males and females? How do these tap patterns vary across species and genera? And do these different patterns reflect ancestry? In other words, are the different patterns synapomorphic? So to do this, we conducted fieldwork in South Africa in January this year. So here on this map, you can see the route we took over 2 weeks. We covered a lot of miles, but this helped us sample a wide range of geographic localities. And then I used a marshmallow piezo contact mic and a recorder with a stereo mic to record the tapping behavior of the beetles. The beetles were kept alive in Tupperware containers and fed oats and apples. And they were kept separated by locality, species, and sex. Fortunately for these species, in most of them, the males have this settle patch on the abdomen, whereas the females do not. So this makes it really easy to separate them by sex. So sort of the initial observations I found, I only observed members of Molarina tapping, so I didn't find any species outside of the subtribe that tapped. The males tapped a lot more than the females. So in the evenings, a few males would get going, and then it seemed like all of the males would start tapping. So they'd kind of get each other going with the tapping. But I think I observed females tapping maybe once. So I suspect perhaps because I didn't separate the males and females right away, a lot of them had the opportunity to copulate. And so I suspect that it may be females won't respond to males after they've already copulated. I don't know for sure, but that's my suspicion. Whereas the males are obviously going to keep looking for mates. It'd be interesting to actually test that to see if that's why the females weren't really tapping. But I did find among the males, I found at least 4 different tapping patterns across the species. So here, the first type I found was a slow tap. The next was a faster tap. This next one was even faster. So this species is tapping at about 6 taps per second, so really fast. And then this last one is they kind of mix it up a little. This is the one I'm calling a ball drop. So you can see they kind of trail off at the end with a faster tapping pattern. So it's sort of reminiscent of the sound a ball would make if you drop it. But this one's my favorite. So now that I had these sort of different categories that I could put the species into, I wanted to map those traits on the phylogeny. So after recording the specimens, they were preserved in 100% ethanol. And then when I extracted them for DNA once we returned, and then using targeted enrichment sequencing, we sampled 628 loci and 700,000 base pairs. So these species were added to this phylogeny I kind of already had. So in total, I have 115 Sepidiini taxa within the phylogeny, and included 8 outgroup species. I aligned the sequences in MAFT, and then I analyzed them using maximum likelihood and IQ tree. They were partitioned by gene, and then used Model Finder to select the models for each partition. And then to determine nodal support, I used ultrafast bootstrap with 1,000 replicates. So here I'm just showing you the clade for Molarina, not the rest of the phylogeny, since that's what we're focused on right now. But I did recover Sepidiini as monophyletic as well as Molarina. And all of the unlabeled nodes had 100% bootstrap support. And so I've indicated the ones that had less than 100, but most of them were pretty high. So first, looking at kind of the clade in the top half of the phylogeny. So this is Maria Zofia and closely related genera. So in this clade, we only had 2 different tap patterns. We had the kind of slow tap and the fast tap. So the 2 kind of straightforward tapping patterns. But you can see both of these patterns are showing up multiple times across the phylogeny. So they're not synapomorphic. They seem to be appearing multiple times, these same kind of tap patterns. And then looking at the bottom clade, so this has mainly Acnodes and then a couple of related genera. So down here, we have this Tibiacnodes species with the really fast tap. This is the only species that I found that had this tapping pattern. And then within Acnodes, most of the ones I sampled had this ball dropping pattern, except for, and this is the weird thing, in this clade, which has species, which Scrobicollis or some similar species, 2 of them had this ball dropping pattern, and then this other one had kind of a more straightforward fast tapping. It didn't trail off at the end with the ball dropping pattern, which was really odd to me since it appears both morphologically and phylogenetically to be the same species. These 3 were all sampled in 3 different localities, but they were relatively close to each other. So I don't know if this is just one aberrant individual, or if there's some sort of population level differences happening with the different tap patterns. So here I have a video of basically 2 individuals from the same species that were collected in different localities. One of them has the ball dropping pattern, and then the other one has that other just sort of regular fast tapping pattern. So it sort of sounds like he's almost going into that same ball dropping pattern and just sort of cutting it off early. And I don't know if this is just this one, because I never got the other 2 tapping. So I don't know if it's just this one individual at this locality, or if others in that same population have the same pattern. But yeah, it seems like it's just sort of taken that regular ball dropping pattern and like cut it short before it sort of trails off. So the last thing I did was I mapped out all of these species that I got tapping data for. I've color coded the same way as I did on the phylogeny. And one thing I found that was interesting was that within a given locality, you didn't really find 2 different species with the same tapping pattern. So here you've got a slow tapper and a fast tapper. In this one, you've got an Ocnodes with the ball dropping pattern, and then another one with just the slow tapping. Here you've got 3 different patterns. So there's that one with the really fast tapping. and then slow tap and the ball dropping pattern. So the only one where there appears to be some similar tapping patterns is these ones I collected around Pretoria. But this includes that Ochnodi species with the weird, where it seems like it should have the ball dropping pattern, but it doesn't. And that does still kind of differ from this Amianthus that I sort of categorized as having the same tap pattern. So it seems like possibly these species are sort of partitioning or sort of being driven to have different tapping patterns so they're not conflicting with each other during mating. But I don't really know for sure. And it'd be interesting to get more sampling to see if that pattern kind of holds. So to conclude, I found at least 4 different tapping patterns across species, and these patterns are not necessarily synapomorphic, so they weren't really necessarily based on ancestry. It did seem like, within a clade you only had a few tapping patterns, but they would be popping up in multiple different lineages. And then we found one case of variation in tap patterns within a species, whether this was one individual or if this is differences between 2 populations, I don't know. And then we found that there were different tap patterns among species in a locality. You didn't really find 2 species with the same tapping patterns. So future research questions I'd like to address. Are there additional tap patterns I haven't sampled yet? Are there differences between males and females? I wasn't able to answer that with this study. Can the beetles differentiate between males and females within their species? And how good are they at differentiating between species based on the tapping patterns? And then are these tap patterns drivers of sympatric speciation? So I'd like to acknowledge my funding sources, the National Science Foundation, the Coleoptera Society, the Society for Systematic Biologists, and the Museum of Comparative Zoology for funding this study. And I'd also like to thank the country of South Africa for providing me with the permits to do this research. And I'll take any questions now.